Students, in this tutorial, we will be studying about the chromosomal theory of inheritance. Before that, a little bit of history. Although Mendel published his work in 1865, his work was unrecognized for a long time. In fact, up till the 1900s, his work was not recognized. And in your book, they cite several reasons for that, the first reason being lack of communication. So today we know that scientists are always communicating and sharing knowledge, but back then that sharing and communication was very less. Second reason was that many of his contemporary and fellow scientists did not accept this concept of genes and factors just because they could not imagine and they could not see what is a gene and factor, and Mendel could not show them what an actual gene and factor was that he was talking about. And also, he had no proof for the existence of factors at that time. So he could not show them and say, okay, this is a factor and this is the proof that there is something called a factor. Another reason, which is also a very important reason, is at that time there was no integration between subjects and sciences. So the fact that Mendel used mathematics to explain biology was not accepted by his contemporary scientists. So these are some of the reasons cited in the book why his work was unrecognized till the 1900s. Now, in the 1900s, what happened? In the 1900s, we had many scientists, okay, and some who are named in your book are Derives, Correns, and Shermark. So these people independently they rediscovered Mendel's results on inheritance, because by that time, that is by the 1900s, there was always already an advancement in microscopy. So people had already taken a look at the nucleus, they had seen the chromosomes, and they were aware about, and they knew how chromosomes behaved. Okay, so at that time it was easier for them to rediscover his work. So what they did was they they rediscovered his work, and there were other scientists, two names given here, Walter Sutton and Theodore Boveri, who noticed that the what they were talking about, that is factors and genes, what Mendel had originally talked about and now they were talking about, they said that their behavior is very similar to that of chromosome behavior. So they found a lot of parallel in the behavior of genes that these Derives and Correns and Shermark were talking about to that of chromosome movement. So they investigated more, and they found that indeed the behavior was parallel to the behavior of genes. Behavior of chromosomes was parallel to the behavior of genes. And they finally, when they were convinced, they argued that the pairing and separation of a pair of chromosomes would definitely lead to the pairing, to the segregation of genes or factors, because these genes or factors are carried on the chromosome. So, what they did was they united this knowledge of chromosomal segregation with Mendelian principles, and they called it the chromosomal theory of inheritance. So, it was Sutton and Boveri who proposed this chromosomal theory of inheritance by taking into account Mendelian principles, because they found that what Mendel talked about genes of factors had similar behavior to chromosomes, and they put two and two together and they said that if chromosomes and genes show similar behavior, that means the genes are being carried on the chromosomes, because you cannot see a gene but you can locate a chromosome in the cell using a microscope. So they assumed that the genes were on the chromosomes, and that when homologous chromosomes separate during gamete formation, you have the genes also segregating and separating along with them. and fertilization restores the chromosome in the diploid condition that we know today. That means when two gametes fuse, you have chromosomes coming back together, and you get the diploid number, and you have the genes coming along with the chromosomes. Now, in your book, there is this diagram where it is shown in the independent assortment of chromosomes is shown. So the possibility 1 and possibility 2 is shown. So in the first possibility, as you can see, is that you have two sets of homologous chromosomes. So you have this homologous pair and you have this homologous pair. So we know that gametes are formed by a process of meiosis, and meiosis has 1 and meiosis 2. So in meiosis 1, you know that it is first what is separating is your bivalent separate. This we had studied in the 11th. So you can see the bivalent separate, okay, in the two cells here. And in meiosis 2, what happens is your chromatids are separating. So you can see here the orange is going along with the green chromatid. Okay, so this is chromatid of one chromosome, this is a chromatid of one chromosome. Okay, so possibility 2 is if this alignment was different, that means instead of orange being close to a green chromosome, the orange chromosome is close to a red homologous chromosome. Okay, so this pair of chromosomes. Okay, so it depends on how your chromosomes are aligned. So here the pair is aligned in such a way that green is closer to orange. Here the pair is aligned in such a way that red is closer to orange. So ultimately you'll see after meiosis 1, meiosis 2, in the gamete or the germ cells, you find that you will find this possibility where an orange and a red chromatid have finally ended in a gamete cell. So this is showing you that it is not fixed. Okay, so it is independent assortment of chromosomes depending on how the alignment is. Okay, what chromosome happens to be close to which chromosome. Then this name is not to be forgotten, Thomas Hunt Morgan, because he did the work of giving the experimental proof, okay, experimental verification for the theory that Sutton and Boveri proposed. So Sutton and Boveri proposed the chromosomal theory of inheritance. Morgan provided the experimental proof. And for his experiments, he used the organism fruit fly. Scientific name is Drosophila melanogaster. As you can see, this is Drosophila melanogaster. This is the female and this is the male. So why did he use Drosophila for his experiments? So he had several reasons. First, it is suitable for genetic studies. Second, it can be grown quite easily on simple synthetic medium in the labs. They complete their life cycle very fast, that is about 2 weeks. Third, in a single mating, you can produce a large progeny, so there is no shortage of flies to experiment with. Then, another very important reason, a clear difference between male and female. So, as you can see here in the diagram, the male is larger than the female is larger than the male, and also you can see that the male has a darker abdomen and a much rounded abdomen as compared to the female, where the abdomen the edge here of the abdomen is pointed, and it is uh, much lighter as compared to the male. So quite visibly externally you can tell between a male and female. And another reason he chose is that even with a very low powered microscope, you can see many hereditary variations. Like some were with brown bodies, some were with yellow bodies, some had red eyes, some had white eyes. So, all that was quite clearly visible. Okay, so this will stop at this, and in the next tutorial, we'll take up the actual experiments and his findings. Okay, so any questions you can post in the chat box.